I'll just really just dive right in. Um, to get things started, I do want to sort of get us all on the same page about how we think about species in evolutionary biology and in plant systematics these days. Um, I think the field has really moved into sort of a process oriented view of not just what species are, but how they are through time. And I think a really nice, albeit simplified, perhaps um, sort of uh, diagram that depicts this is what we see here. So this is from a DeCuro's paper. And here we have this idea of at some point in time there is a single lineage, and maybe there are multiple populations of species in that lineage. And as time goes by, as we move up this diagram, different things happen to these populations. They acquire different characteristics that begin to distinguish them. And that's what we term sort of that gray zone of speciation. And as time goes by, as more and more of these characteristics are acquired, at some point in time, we have a very distinct set of populations that we recognize as different species. But sometimes we are encountering these populations early in this process, when just one or maybe two criteria have been acquired, and it's much more difficult to distinguish species. And one thing that I like about thinking of this process and thinking of how we are in this process is that it, of course, acknowledges that speciation is a continuous process. It's always happening. And especially in the early stages of speciation, any kind of criteria really could diagnose species. And so the job of the modern systematist is to identify what are those criteria, and when in this process are we sampling them. And we use lots of different types of data to do that. Certainly molecular data is really
All right,
important,
so running.
but also morphological, ecological, et cetera. So as a systematist, my work is really geared towards Castilleja, like we saw in my title slide. And one of the great things about Castilleja is, of course, they're very beautiful. I think most people think of Castilleja as something like this, these big fields of bright, showy red flowers, sort of with beautiful mountain landscapes behind. And many of them do occur in these really beautiful areas, but they're not all red. They don't all look just like this. Here's a collection of photos that I think illustrate a lot of the really beautiful shapes and textures and colors and stuff that we see in this group of plants. There's around 200 named species that are distributed widely across the Americas. And this is a group where taxonomy and systematics has really been challenging through time. I think many folks recognize this as a challenging group to identify an individual to species within. Most people who have worked and published in the group, they comment on it in some form or another. And for those of you that have seen my talks before, I'm really sorry, but I have to tell this joke again, or I have to share this one quote that I just think is so perfect. So this is a quote from Harold Rickett, who did the wildflowers of the United States. And in one of his sections, the northeastern United States, he says, "The genus Castilleja is one of those that make botanists wish they had embraced some easy branch of science, such as theoretical physics. It is a Western genus with at least 250 species, on the exact characteristics of which no two botanists seem able to agree. Fortunately for the readers of the present volume, only 4 species are found in the northeastern states, and they are not hard to distinguish." I think it just really nicely encapsulates sort of the challenge that most folks have when encountering this group. So my talk today is going to sort of cover 3 main elements. I want to sort of dig in a little bit to why taxonomy is so challenging in Castilleja. I also want to share with you how some of the really data-driven approaches to trying to delimit species in a handful of species complexes have yielded some insights, but also revealed more questions. and then share with you sort of my anticipated approach towards trying to understand what species in Castilleja are. So to begin with, why is taxonomy in Castilleja so challenging? I think it boils down to a couple of sort of primary reasons. So first of all, you can find Castilleja in lots of different places. They have a very broad ecological amplitude. You find them in wet areas like Castilleja cusickii from the mountains of, or the meadows of the Intermountain West. You find them in dry areas like Castilleja scabrida from the high deserts of Utah and western Colorado. You see them at high elevations, or I'm sorry, low elevations like Castilleja latifolia from the coastal sand dunes here along the California coast, to high elevations like Castilleja nana from the subalpine to alpine areas of Sierra Nevada and the mountains of Nevada. We also find them in very extreme environments like Castilleja salcigenosa that occurs in these really harsh alkaline soils in Nevada. And among all of the different named species, there are some that are very broadly distributed across very large amounts of geographic space, and others that are more narrowly restricted. So you encounter them pretty much everywhere you go. But they also have biology and physiology that I think has a lot of still sort of open questions about how that contributes to taxonomy and its challenges in Castilleja. So Castilleja are hemiparasites. They're capable of performing photosynthesis. They form an organ called the haustorium. So this is a really nice histological section of a close relative called Striga. So Striga is, sort of here on the left side attached to a host, and in the right we can see the tissue of the parasite sort of punching a hole into the host root, and you can see that it's actually accessing the xylem and the phloem here and pulling out water and nutrients and other types of things. We sort of make the assumption that they are entirely facultative, although there have been some hypotheses that there might be obligate forms. This means that they can survive without a host. So this is a picture of Castilleja scabrida that I took where that rock extended far beyond the frame of the photo here without another plant in sight. This organ, the haustoria, is this very sort of delicate structure. This is a picture of some tiny little seedlings that are germinating on a penstemon, I believe. And one thing that makes studying the actual host of any given individual is that those haustoria are very small and those connections very delicate. They rip apart very easily when you try to remove a plant from the ground. So the role of parasitism in the evolution of Castilleja, I would say, is very poorly understood. Another reason why taxonomy in this group is challenging is because they have a very sort of complex molecular evolution. And there's a couple of elements of this, and I'm going to summarize them very quickly here, but I'm going to dig into them in a few slides. But from a molecular perspective, this group is very young, so we estimate probably somewhere between 4 and 6 million years old. It's also a group where we regularly see evidence of gene flow, especially when species co-occur. And this even happens in things that we think are putatively very distantly related, so things that have very different morphological forms. So a great example of this is the White Mountains in the southern sort of eastern Sierra, where we find Castilleja linearifolia, which has a very long-beaked, long-flowered morphology. We see it in the same population as Castilleja, there we go. Castilleja pilosa, which is a much shorter beaked, a shorter flowered form that we think is probably more bee pollinated or fly. And in those same populations, we see these really beautiful sort of phenotypic intermediates that are, right next to both Linearifolia and pilosa. So we frequently see evidence of this. And then another piece that makes the molecular evolution of Castilleja complex is the fact that it is a polyploid. And that polyploidy is really widespread across the group. So polyploidy in Castilleja is something that many people have pointed at as contributing to taxonomic complexity in its own right. Larry Heckard and T.I. Chuang did a huge amount of work to try to understand sort of the breadth of the incidence of polyploidy and sort of the levels of ploidy that exist within species. They have, counted chromosomes in, hundreds of different species. And in the case of more than half of the 100 species that they counted, these were polyploid or they were diploid with one or more additional polyploid levels. And so here I have a map of just the majority of those that have been published in their papers with the colors corresponding to the different ploidy levels. And in a second, I'm going to click to the next slide, and it's going to show you these different ploidy levels one at a time so that you can see them individually in their geographic breadth. And the reason for this is because, sometimes we look for patterns of where these ploidy levels occur, trying to understand things that might be correlated with polyploidy or correspond with them. And what Heckard and Chuang found was that actually there might be patterns there, but they're very difficult to tease apart, and probably we should be measuring more individuals. They also have a word of warning in one of their papers that sort of points at the fact that so many of these species have been measured at different levels. They looked at Linearifolia, I believed, and had measured 20 or more individuals, all at the diploid status, and then their 21st, 22nd, 23rd individuals were all polyploids. So I certainly took that to heart to suggest that, the ploidy status of any perennial species that you're encountering really does need to be estimated because it is so widely variable. And in all of their papers, they really do point at hybridization and polyploidy as being suspected to contribute to the taxonomic difficulty. So in this idea of polyploidy and hybridization has been empirically tested in a number of different cases, and I think some of Erica Hirschgreen's papers are particularly good at digging into this. So she looked at a handful of species that were occurring in the Copper Basin of Colorado. She was in this particular basin, there were, I believe, three different species. So here, panels A, B, and C are the three species that were there. And panels D through H are all sort of intermediate or variable forms that she was finding. And so in a series of 2 papers, she was estimating the ploidy of all of these individuals, and she was also trying to understand if gene flow was happening across all of them. And from this Tangled Trios paper, the major take home was that hybridization was definitely happening across different ploidy levels. It was certainly possible, but probably less successful across multiple ploidy levels. She also found that some of these morphological variants were in fact hybrids and might be the result of sort of older stabilized hybrids. She found that interspecific gene flow was happening in all directions, but some of the barriers were weaker. So conspecifics, it was definitely easier for introgression to happen, whereas when you were going across species, it was less successful, but certainly possible. I think I said this just a second ago, but species were nearly fixed for their different ploidies, but some deviations did occur, primarily in these populations with variable plants. And then, she found that interspecific gene flow could occur across all these ploidy levels, more likely with the same number of chromosomes, but it was possible in these others. And so I do think that this is just a good reminder that we can't discount polyploidy and the complexity of this group, and so much of my work is trying to account for that. So I'm going to shift gears a little bit and talk about how we currently sort of conceive of the organization of Castilleja, and how that has sort of influenced what we think the major relationships in this group are. So here I have a series of diagrams that sort of illustrate the major morphologies that we find in the genus Castilleja. Underneath each of these morphologies is a box that tells you the number of sections and species that correspond to that particular morphology. And if you look pretty closely, you can see that a couple of these morphologies are fairly novel or unique, with only one species assigned to them. And so really the bulk of the morphological diversity is found in these 5 primary morphogroups. And our early sort of assumptions of how these things were related to one another took that into account. So this is a modification from Chuang and Heckard's paper in the early 90s. And you can see that some of these less speciose forms, these more unusual forms in Castilleja, they hypothesized to be sister to the rest of Castilleja. So Clevelandia, Ophiocephala, Gentria was considered part of Castilleja in their treatment, but they're all sister to the rest of Castilleja. And one thing to note here too is that the annual group this section here, Oncorhynchus, was thought to have derived from a perennial background. And this is a very common hypothesis about the evolution of annual habit from a perennial one. And in the sort of mid to late 2000s, Dave Tank started applying molecular phylogenetics to this group, and he estimated the first molecular phylogeny of Castilleja, the first phylogeny to begin with. And one thing that was really cool about this tree, or this inference, is that we begin to understand that there is a lot of morphological variation in Castilleja, because all of these sort of novel, unique forms were actually nested within Castilleja as a whole. The other big thing that sort of shifted once we started applying molecular techniques is that we understood that the perennial habit of Castilleja was really derived from this annual background. So perenniality deriving from a most likely annual common ancestor, which was a big shift in the thought of the evolution of this. But this work only sampled, had limited sampling, both in known species, but also in parts of the genome. And so when I came into systematics of Castilleja, one of my major goals was to increase the sampling, both across the genome, but also increasing the sampling of the species. And so my work really focused on developing primers that would help us amplify certain parts of the chloroplast and the nuclear genome. And so these were primers that were specific to Castilleja. And we pretty quickly realized that polyploidy was sort of throwing a wrench in our nuclear data. We had tried to design primers around parts of the nuclear genome that were presumably in single copies. and yet we were recovering what we thought were multiple copies, which suggested that there were multiple copies of the genome, and therefore probably we were dealing with polyploids. We didn't necessarily know the ploidy status of these individuals, and so we really, during my dissertation, I focused a lot of my effort into trying to just use the chloroplast data. And so what you're looking at here is a phylogeny of using just the chloroplast data of this increased sampling of Castilleja. And this reconstruction has approximately 200 species in it, with many individuals per species. So this tree has almost 450 tips. There was high backbone support for the major clades, with much sort of more minimal support at the shallower nodes. When we started examining this tree and trying to understand what these patterns would tell us, the first thing that really popped out to us is this really impressive amount of phylogenetic geographic structure. And so here, instead of putting the names of the tips on the trees, I'm instead color coding their position in the tree based on where they are on the map. So all of the individuals that are in this top clade each got a red dot, and then I put them on the map corresponding, giving them the color that they were assigned in the tree. And I did this for most of the major clades in the group. And when you look over at the position of these individuals or these clades in geographic space, we can see that there's just a lot of structuring here. And this is a really sort of impressive and very strong signal of this sort of geographic structure. When we start sort of digging into what these patterns tell us when we take the identification of the individual at the tip of the tree into account, things become really interesting. So first of all, widespread species, for example, like Castilleja miniata here, are found in multiple clades. So we can see from the map that this species is widely distributed across geography, and it is correspondingly widely distributed across the tips of the tree. So this is sort of a freaky feeling when you see, things that you think of as a cohesive species sort of popping up in multiple places of the tree. When we zoom into a particular clade, we see that these members of the clade that have been resolved as putative close relatives are composed of many different morphological groups. So here is one clade that has species from multiple different morphogroups, multiple sections within those morphogroups, and all sort of clumped together. If we look at these morphogroups and try to ask if they are sort of reconstructed to be closely related in this phylogeny, we find that that is not the case. And this is just one example here where this widespread morphology is again found all across the tree. And so if we try to summarize some of the take-homes of what this phylogenetic geographic structure is telling us, the first thing is that our taxonomy is at odds with the chloroplast phylogeny. They're just at odds. This also suggests that morphology, just sort of generally, and the molecular signal of relationships in this group may be in conflict. And I guess, keep in mind, this is just a chloroplast phylogeny, and so ultimately we don't really believe that this is happening, that this is, indicative of the primary relationships in Castilleja, but it is beginning to give us a clue about the sort of just complex history of this group. So this is sort of where my dissertation, from a genus-wide perspective, where my dissertation stopped. my other chapters were starting to work in species complexes in the group, and then I went on to do a postdoc and ultimately to start my research lab. But Dave and some of his students continued this effort of trying to understand and quantify the molecular evolution of the group, and they did that by really beginning to dig into the nuclear genome in a much more holistic way. And so the way they did this was by applying the angiosperm 353 probes to this question of evolution in the group. So these probes are what we call universal primers, and they're designed for all of the flowering plants, and they amplify conserved areas of both the chloroplast and the nuclear genome. So this was really cool. They could look at the nuclear genome, but also actually sort of see the signal or look for this signal of geography with the chloroplast reads as well. But ultimately, this was just a really nice way for us to begin to access evidence from the nuclear genome. And so this was work that was led by Malia Santos, who was a grad student in Dave's lab and is now a postdoc actually with Stacy Smith. And so this is some of her work. When she sort of inferred, she, sort of dealt with the issue of polyploidy in these data. You're able to sort of sort the paralogs and inferred a tree, a phylogeny for this group. We continued to find this geographic structure, albeit in a slightly less intensive way, where North American annuals are sister to everything else. So that's these guys down here. And all of the perennials are sort of derived from this annual background. So that's consistent with patterns that we had seen previously. There is still some geographic structure. We do see that the North American perennial clade, this large clade up here, corresponds to this sort of light blue color that is in the map inset. We see the South American perennials and the Mexican perennials resolved as clades as well. And then we continue to see some sort of mixing of species complexes that we would otherwise expect to be closely grouped together. So for example, the Pilosa complex, we have members in multiple different clades, all within this sort of North American clade here. And I'm going to talk about the Pilosa complex a little bit more. So we continue to see a bit of this geographic structure, the signal of what, from a taxonomic perspective, what one would expect to see, cohesive species complexes still tend to be widely scattered across the phylogeny. So because we have these 353 different regions, we can start to ask questions about the consistency of this signal in these different parts of the genome. And so we do that by asking how concordant these different genes are with a phylogeny. And so Malia performed this concordance analysis. This is done with a package called, PhyParts. And here you basically take each one of those 353 genes, you infer a phylogeny from them, and you compare them back to a species tree that you've made. And at every single node in the tree, you're asking how many of these different gene trees are in concordance or are perhaps discordant with the species tree topology. And so the way you read this is by looking at these pie charts at each of the nodes. And the colors correspond, the colors of the pie correspond to gene trees that are in support of the topology in blue or are supporting other topologies in red or green, or potentially really just have no information about that particular node in gray. And so if you have a highly concordant set of gene trees with a species tree, you would see a lot of blue. And as you scan these nodes in the phylogeny, or in the species tree, you can see that we actually have quite a lot of red, which suggests that there's a lot of conflict in this data set. And generally speaking, we see that really the only concordance is at some of these deepest nodes, that blue color, remember? There's very minimal amounts of concordance in some of these geographic clades, and generally speaking, just very little concordance, except at some of the most shallow nodes where we begin to pick up a little bit of support for this species tree topology. And so I think a very reasonable question is, why is there so much discordance with this phylogeny or with this species tree? And so Malia really applied some cool techniques to examine this question. First of all, she did a series of simulations to try to understand if this conflict in signal was a result of incomplete lineage sorting, and sort of just the nature of how different parts of the genome will evolve at different rates. And her simulations suggested that that was not enough to explain all of the discordance that we could see, or that she could see in the data set. And so she began looking at hybridization as a contributor to all of the discordance. And she did that in a cool approach called Tinnick. And well, it's a combination of approaches, Nanook and Tinnick. And ultimately, the result is a network that sort of illustrates the sort of quantified hybridization that is found in the tree. And so this figure here is a representation of that hybridization network. And we've got some of the same colors that corresponded to those major geographic areas. So this light blue color here corresponds to that big sort of North American clade that we saw on the previous slide. These are the South American and Mexican clades, and then we've got our annuals here. And I think one thing, so in these networks, all of these sort of cross lines between the primary branches that are going out to the tips of the tree indicate areas where there is a suggestion of hybridization. And so you can see that this hybridization has occurred across many different, if not every single branch in this network. And we're seeing it not just with things that are very close, but also across very broad areas. And when we consider the geographic distribution so now I'm looking at the color of the tips of the trees here, we can see that there's some really interesting geographic substructure within that big North American clade, for example. So all of this is to just say that there has clearly been a lot of hybridization in this group involving many of these different tips. And this was, a pretty impressive amount of of hybridization, actually. So, the work, I think, Malia's work is really illustrating that the evolution of this group is clearly very complex. And given the very young age of this lineage and the extent of hybridization that's happening throughout the network, we're proposing that hybridization has probably been very cyclical and has been repeated multiple times throughout time, since this lineage diverged. And we think that we also have pretty strong evidence to suggest or to propose a hypothesis called the multi-compartmentalized syngamian hypothesis to try to explain this geographic pattern. And so these two insets on the left side illustrate that at different times during glacial cycles, these different clades, these different sort of geographic clades are either in isolation, but mixing within, or they're connected and mixing among. And if that type of cyclical sort of mixing happens over and over and over again, we think this is what could lead to that extensive amount of hybridization. So this work, Malia is in the process of finalizing this manuscript for submission, and we're really excited to see it published. And once it's out there, I hope that you all will go and check it out. It'll do a much better job than I just did of explaining some of these analyses. But this framework, I think, really indicates to us how much hybridization is playing a role in sort of the challenge of understanding the primary relationships in this group using molecular data. So I'm going to transition now into sort of my work with species delimitation and how I've been trying to use really data-rich data sets to understand where species boundaries are. So right now, if we, try to identify an individual to a species in this group, we run into a couple of tricky areas when using keys. And I have a couple of slides here to sort of illustrate this challenge for those of you who haven't, tried to identify individuals to species in this group. So most taxa, especially those within species complexes, really overlap in quantitative traits. So here I'm going to walk through an example of a group of species that I'm working with now, the coastal California Castilleja complex. And so I've got a suite of pictures here that sort of illustrate the similarity of these species. And then down here I have the major floral components of Castilleja, and we are oftentimes taking linear measurements of these floral pieces, but also the height and the inflorescence characteristics as well. Now, as I say, many of these sort of linear measurements, these quantitative traits, heavily overlap within the case of closely related or very similar looking species. So as an example here, I'm showing you the corolla length of the members of this C4 complex. And I'm purposefully not telling you which line goes with which picture. But the minimum and maximum values are taken from the species descriptions. And you can see right away that the minimum and maximum value of the corolla length is widely overlapping across all of these individuals, or across all of these taxa. And so if you took a sample of individuals that might have been assigned to this pink species and put the raw value of the length of the corolla, it could technically, of course, apply to the pink species, but it could also apply to any one of these other species. Now, this is just a single trait. We often don't use just single traits to identify species. And so if we add another trait here, for example, the length of the beak, we can make these sort of areas of expected corolla length and beak length size that we might see. And so if we add the beak length to each of those individuals that we saw in the previous plot, it might look something like this. So now we have individuals that have been assigned to this pink species that are actually falling within the expectations of all of these other species that are part of this complex. And so I think this illustrates for you like sort of how difficult even just two characteristics might be to try to identify species. And we see this with that pink species, but also the green, the other green, the blue, and the pink. They all just widely overlap with one another. If we toss all of these quantitative measures into a principal components analysis, where we sort of let all of that information contribute to how to characterizing how similar these traits are, we get something that looks like this. So here I'm using a suite of, I think it's 15 different linear traits to describe this morphospace. Individuals are color-coded according to the species that they were assigned to, and then plotted here. And these ellipses, show you where the primary concentration of these individuals are. And so across all the different combinations of the first 4 PC axes, we see just a lot of overlap. We do see this quantitative overlap in other species complexes as well. So I have data sets in members of the Castilleja species complex, where we have sort of our main players are Castilleja and Ludia, and then also this other individual, Pulchella, that looks quite a lot like Castilleja. And so individuals have been color-coded to those species, and we can see in the 3 axes, or the 4 axes of the principal components analysis, that they're quite overlapping. Certainly along PC1, we can see that Pulchella is inhabiting this sort of unique corner of the morphospace, but Ludia and Castilleja are very much right on top of one another. Another data set that we have, or another species complex that we have a lot of data within, is the Pilosa species complex. This particular group has a number of individuals, or a number of varieties named within the species Pilosa. There's also Castilleja nana, Castilleja salsuginosa, and here again I've color-coded individuals to these different species. We plot them in PC space, and we see just a lot of overlap. A little bit of distinction for some sort of areas, but just a lot of similarity. So these quantitative traits sort of break down for us in many ways, and so we also rely on qualitative traits to diagnose species, but these are difficult at discriminating taxa as well. So my example here is in the coastal the California coastal complex. We've got primarily red taxa, but each of them have a morphological variant or a color variant associated with them. One thing that I'm really discovering here as I'm doing fieldwork in this group right now is that within species or within population, color variation is very common in this group, and it is not uncommon to see suites of individuals that are along this incredibly wide spectrum of colors. So it's not uncommon at all to find very pale yellow individuals all the way up to bright sort of crimson red individuals. We see some of these qualitative characters rarely diagnosing or discriminating taxa in the other 2 complexes as well. So in, let me go back. So in our Castilleja complex, we have historically really identified Ludia by these darkly contrasting veins that we see in some individuals. So as a reminder, this is the PC space using those morphological traits that we saw earlier. Pulchella is here, and Castilleja and Ludia are largely overlapping here. And in this next plot, this is the same morphospace, but instead of color-coding individuals associated to their species, I've color-coded them according to whether or not they have these dark contrasting veins. So if they do, we say yes, and if they don't, we say, if they do, we say yes, and the color is yellow, and if they don't, then we say no, and the color is this green. And you can see that the yes and no, the with or without these contrasting veins, is widely dispersed across this morphological space, and individuals of both Ludia and Castilleja have these different colored veins. In the Pilosa complex, one of the qualitative traits is the presence of a dark colored spot on the corolla. So sort of just as we just looked at in the Castilleja complex, I've got the normal sort of PC space here with individuals color-coded according to species. And when we plot those same individuals, but now color code them about whether or not they have this colored spot, we see that the yes or no, the yellow and purple, are sort of mixed together across the Nana and Pilosa area where we would expect to see this discriminated. And so these qualitative features sometimes don't help us either. And so we often lean on geography as a real driver of diagnosing species in this group. And when we look at some of the floral features of members of the coastal complex, we would technically, using the key, identify this particular individual as littoralis. But these three individuals here, we would also assign to littoralis if we ignore geography. If we take into account geography, they're considered affinis. So their morphological traits, assign them to littoralis, but it is geography that says that littoralis is actually restricted in the northern areas, and affinis being primarily on the coast. And so I don't point these things out to say that the keys are wrong or anything like that. It's just that there's so much sort of conflicting signal that is inconsistent. And so when you're in a given population, it's so hard to know, am I in a population that has this colored spot, or am I in a population that has these veins or not? And yeah, it's just really challenging. I think one of the important things that sort of I've come to terms with here is that we don't understand the mechanisms that are underlying these traits. And so a good example here is in this Castilleja complex, where I think we have evidence of sort of a change in morphology over an elevational cline. So here I have, again, the morphological traits. Remember that Pulchella is here, Castilleja and Lutea are here. And you can see the elevation that each of these individuals was collected at in this lower graph. And these individuals were all at lower elevations. They're assigned to Castilleja and Lutea. And as you move into higher elevations, we start calling things Pulchella. When we look at actual measurements associated with these different taxa, we see this cline in effect. So here I'm plotting elevation along the x-axis and the width of the raceme along the y. And then the shape here is associated with the species that it belongs to. And then the color is with elevation. So it helps to highlight this thing. And we see that as the raceme width gets narrower and narrower, we're finding plants that are at higher and higher elevations. Similarly, if we look in the Pilosa complex, I think we see a latitudinal pattern associated with that corolla spot that we talked about, or that I mentioned earlier. So here, all of the populations that do not have this corolla spot are almost entirely north of the 41st latitude. And most individuals that don't have the corolla spot, regardless of taxonomy, that do have the corolla spot are at this southern latitude. And so this really suggests to me that there are other elements at play here driving some of these characters. And that really sort of makes one begin to think what we're naming when we put names around these things. And so here I have a meme to sort of point out that, we are assuming that these morphological traits correspond with species boundaries, and it is possible, at least, that some of these traits might be plastic. And so some of the things that we are looking at to help us try to diagnose what species are might in fact be environmentally regulated or anyway influenced. And so a lot of my work in these complexes has really tried to collect extensive morphological data sets that are one-for-one complemented with a molecular data set. So I want to have for every single individual some morphological data, some molecular data, and begin to compare and contrast the different signals of species boundaries in this group. And so this has happened now with both the Castilleja and the Pilosa complex. So I can share with you some of these patterns that we're seeing. So from, just to remind you, from this morphological perspective, we are seeing, I think, this elevational cline in some, but not all of the traits. So here are three different morphological traits. One of which you've already seen has this strong elevational component, but two others that are completely agnostic to it. And then when we start looking at the molecular signal in this particular complex, we see a fairly chaotic mess. So this is one of the first RAD data sets of species complexes, and this was put together by Caroline Brose, who's one of Dave Tank's students. And here we have kind of like what I was showing you with the chloroplast phylogeny. We have, or she has color-coded each of these primary clades that are inferred from the phylogeny with a particular color. And then she's put them in their corresponding spot on the map and color-coded that point with the clade that it comes from. And I think the first thing that I saw was that these clades do not have geographic structure. They're all kinds of crazy mixed up. When you look at the species names on each of these trees, you see that each of these major clades is composed of all three members of the Castilleja complex. And when we look at their geographic position here, there is no structure. And I think the best spot to see this is in the little southeast corner of Idaho, where we literally have a member of each of the three major clades in close proximity. So this is wildly different than what we're seeing from the clade-based phylogeny that I was showing you earlier in the talk. Now, when we look at the Pilosa complex, we find that a very different pattern. So here we're not looking at a phylogeny, and instead we're looking at admixture results, which also give you some sort of understanding of how genetic variation is distributed across the landscape. And so we can see that the major genogroups that have been identified in this analysis are very geographically structured. So this green group is in the northern latitudes. This yellow one is sort of isolated here in central Idaho. We've got the pink group, and then these two sort of slightly mixed groups in the Sierra Nevada. But when we look at morphological variation, you've already seen some of the PCs that have been built for the Pilosa complex. But we started asking how, one of the novel things that we've done with this data set is ask how each of these individual traits may, or the variation in each of these individual traits may or may not be sort of correlated or described by changes in both abiotic and biotic factors. So here we've got all of our different traits along the y-axis here, and a suite of abiotic factors and then biotic factors. So latitude, some major climate variables, and then Katie, who has led this project, was looking at both herbivory and pollinators that were visiting these groups. And then the cells of these comparisons are the products of Permanova analyses, where we're asking about how any particular variable might be explaining variation in these morphological traits. And the darker colors suggest a stronger explanation of those variables. And we can see for a lot of our morphological variables, we've got a lot of dark colors here in these sort of abiotic places. So with these abiotic traits or variables. So that's another sort of indication that that these traits are at least related to or correlated with the abiotic environment. So, I think from a molecular perspective, these two examples really illustrate this fascinating level of genetic complexity. We see this clear geographic signal in one case, but also sort of a nice big mess of confusion in another case. And I think if we frame this in light of what we understand about the diversity or the complexity of molecular evolution across the genus, we have some really great testable hypotheses about evolution in this group. And I really think that these two examples are more evidence of syngamians. So the Pilosa group here on the right has this particular complex spans multiple compartments of the syngamian. And so this particular compartment is behaving sort of independently of this compartment and this compartment, even though there's a lot of morphological similarity that exists here. Whereas the Cusickii complex exists within a single compartment and reflects a lot of the hybridization that's been happening more recently. And so that's what I mean, where these sort of data-driven approaches are yielding more questions, perhaps, than answers. So all of this work is really helping us to develop hypotheses that explain the genomic evolution in this group. But from a morphological perspective, I think things are still really confusing. And much of it centers really on what I think is our lack of understanding of within species trait variation. And so I have a quick conceptual diagram that sort of explains why this is so confusing. So within species trait variation, I don't think that we have a good sense of this. Is this variation centered within populations? Is it among individuals, across populations? Does the amount of variation vary by species? Those are some open questions. And this within species trait variation exists against a backdrop of genotypic variation, where things like polyploidy and drift and mutation and sort of population level things are happening. But it also exists in a backdrop of more trait expression variation by genotype. Things like plasticity, for example. And then, of course, this all sits within the context of environmental heterogeneity. Like my earlier quote, the western species of Castilleja are a hot mess, and they are in an incredibly heterogeneous and very dynamic landscape. And this then creates all of these sort of like microhabitats and stress gradients and lots of different types of communities that these species are existing in that are sort of acting in different ways in different areas of geography. And then, of course, all of this exists against the backdrop of gene flow. And I think we have really solid evidence that there's been a lot of gene flow historically, and probably a lot in very contemporary times, and probably many cycles of this between. And so as I move forward in trying to understand species and species complexes within this group, I'm really trying to build out these data sets that will help us to begin building information about species within species trait variation. And that means sampling more heavily within populations of species and doing that in a consistent way across populations, trying to make sure that at each of these levels we're we're accounting for variation that could be at the biotic, but also the abiotic, influencers in those areas. And then finally, we have to take other potentially co-occurring species into account when we set up this sampling design. And so when different morphotypes or different species of Castilleja are together, we've got to make sure to sample all of the variation and account for that. And I am hoping that a better understanding of the source of trait variation within this group is really going to help us wield this data to better understand what species are in the context of this syngamian sort of evolution. And so I want to walk through, this is my last couple of slides, to sort of help tell you how I'm going to try to use these two different types of data in conjunction with one another. So when we think about these big data sets that I'm building, how we're actually going to bring them together to try to put boundaries on species. And I think right now one of the best things that we can do is let each sort of line of evidence tell us where there is structure in these groups. Let them sort of speak independently and then ask where things are in congruence or not. So as an example, I want to walk through some of the recent data from the Pilosa complex. So our admixture analyses found these 5 different genogroups that have this geographic structure. Our morphological analyses assigned individuals or found this was agnostic to species boundaries. This was just the morphological variation that existed. Best fit a model of 5 groups, and this is what they look like in morphospace. And so I want to show you how we can compare and contrast them. So we can basically build sort of a contingency table where we put, for example, our different phenogroup possibilities along the rows and our genogroup possibilities along the columns. And then for every single individual that we have, we can take its genogroup and its phenogroup assignment and place it in the appropriate cell of this table. And when we do that, I think we can begin to see different types of species that emerge. So for example, so here's that same table. Now I'm sort of identifying the geographic origins or the geographic locations or context of each of these genogroups. And then the 5 phenogroups here. We have, in this data set, we have something that we would call potentially a good species, where you have a uniquely exclusive genogroup and phenogroup pairing. So this genogroup only is associated with this particular phenogroup. And then also in this context, we can identify things that we would call genetically cryptic or morphologically cryptic. So here we've got an example of a genetically cryptic thing. So from a genogroup perspective, it's a single genogroup, but we find that individuals within this genogroup are actually assigned to multiple different phenogroups. And so genetically, it's cryptic, very similar, and they all look morphologically different. And then there's a reverse side of this, the morphological crypsis, where we have a single phenotype that actually has multiple genetic signatures that has been observed within it. And so this comparison, this comparing and contrasting these different types of crypsis is a way that we can begin to describe the variation that we're seeing, but also use this to help guide further assessment and research. And perhaps once we do understand these things a bit better, we can sort of reassess the data with a new baseline knowledge. Now, I think the kicker is, of course, once we recognize these different types of species in Castilleja, we still have to think about how we utilize that. And I think this is a really challenging question. And I don't have good answers for this just yet, but these types of questions are like, what actually diagnoses this kind of thing? And how can we construct or erect a classification and taxonomy in light of these insights? This is a particularly challenging thing, I think. I'm really excited about this idea of what we call like dynamic monographs that allow us to contribute new data very quickly, and these monographs would allow us to potentially absorb and respond to new data that is collected, as time goes by. I think we also have real challenges thinking about how we conserve and manage things that are not necessarily field diagnostic, and thinking about how we conserve or manage a syngamian is also a difficult question to answer. I am really hopeful that the more extensive sampling that I'm trying to employ with these data sets is going to ultimately be useful here. And then finally, how can we sort of communicate this information out to practitioners? And I do think that close collaborations with land managers and conservationists are going to be really helpful for us to figure out what that landscape looks like and how best to do that effective communication. And so that's my last slide. I want to just say that this is like a really complex group, and there's so much work to be done, and I think there's a lot of opportunity for some really cool things to happen, a lot of cool questions and research projects to come out of working in this group. And so I just do want to just ask, if you're out there, if you're doing any of this kind of work, if you work in Castilleja or in similar types of groups, if you would like to coordinate and collaborate, I would love that. So please do reach out and get in touch. And I guess with that, I will end with a picture that I think nicely illustrates what I think evolution in this group looks like, which is very much a braided river approach, where we don't see those strict bifurcating topologies that I think many of us wish that we had, and instead a very hyper-connected group of lineages. So thank you.
Thank you, Sarah. That was amazing.
Well, thanks.
Really sort of, intriguing and terrifying.
Yeah. Yeah.
Because it really puts into question everything we're trying to do. Don't you think?
Yeah, it's scary in some ways, but at the same time too, I think we're just learning about how much cooler plants are than everything else. They really are surprising us in lots of different ways. And with the advent of, or not the advent, but like with how much easier and cheaper it's getting to generate sequence data for many individuals within a single species and ask these types of questions, I think we're going to see this more and more frequently. I think we actually are. And so, I wouldn't, it is kind of scary, but I think it might be a new paradigm shift for us. Yeah.
So Peter Alpert has a question.
Yeah. Hi, Peter.
Hi, Sarah. Thanks a lot for the talk and the quandary. Is there any reason to think that parasitism or hemiparasitism might promote a reticulate phylogeny?
Yeah, that's a great question. So, the role of the host or the parasitic nature in general, I don't think we have a good sense on its pressure for the evolution of the group. So, one thing that comes to mind is there any sort of confusion of signal that might be influenced by the host that we're detecting in the genome? So is there genetic material that's moving from the host to the parasite? And they've seen that in other types or other lineages of parasites outside of the Orobanchaceae, but not to my knowledge has it been seen in Orobanchaceae, and I don't know that I would expect it here. I suspect that Castilleja are generally generalists in their host preference, and so it would be pretty tricky to try to detect that, I think. Yeah, yeah, I don't know. Yeah.
So we have a question in the chat from Ian McFall. Ian, do you want to just unmute yourself and ask it?
Sure. Thanks so much for this amazing talk. I was just asking, I'm in Phoenix, Arizona, where Excerta is our most common species. Can you give any more detail on, I know you mentioned that sort of Mexico and South America grouped together. And like, so from that versus sort of the California and Sierra Nevada and Nevada species, how, like where the line is drawn, if there is a line given this last slide between sort of the North American US group and like, again, where is the US versus Mexico in what the data show?
Yeah, let me see if I can, if you give me 1 second, I can move my slides back. I think I can get us back here. Maybe this slide. So where these seams are specifically, I don't think that we have a good sense of that. So this particular data set is still not incorporating a lot of species diversity. I think there was like 70 species in this tree in a group of like 200 or more. And so I think that with additional sampling, we might begin to see this a little bit more. Our original geographic clades were really sort of seeing a blob from sort of Mexico up into the Great Basin, with the Pacific Northwest sort of cupping around it with the Rockies, and then mostly Southern California and into Baja. And they're seeing slightly different patterns with the 353 data. And so I think that, yeah, it does make a lot of sense to me if we think of like major ecoregions across North America. Of course, it depends on how sort of resolved you want those ecoregions to be, but the sort of bleed of the Great Basin into the mountainous areas of northern Mexico, I think makes a lot of sense. So we also know that those seams, like Sierra Nevada, I think is a great example of a seam between the California and then the Great Basin ones. Those individuals that are at that seam also flip and flop between the different compartments. And so those are going to be particular gnarly areas, I think, to try to understand which Syngamian compartment they have most recently been in or that they are currently a part of. And those are some of the questions that we're really, I think, going to start digging into from the genus-wide perspective in the coming years.
So Andy, since Simpson has a question or comment.
Yeah, I brought this comment up at the very beginning, but it appears to have been skipped over. You said very, very early on that you think that the entire Castilleja species complex, the entire genus, is circa 5 million years, a few million years old. And and your talk with an enormous amount of introgression combined with what I know about a different group, namely the tribe Malayi and the Rosaceae, that rang an alarm bell that I just want to bring up exists. The Malayi, if you do a molecular clock phylogeny on the origin of on the last common ancestor of the Malayi, it's within the last 10 million years, possibly within the last 5 million years. However, there are fossils of living genera in the Malayi that are upwards of 45 million years old. So clearly the amount of gene flow between different genera in the Malayi has resulted in the molecular clock, greatly underestimating the age of the genus.
Mm.
As far as I know, you may correct me on this, Castilleja doesn't have much in the way of a fossil record, but I caution everybody about using molecular clock, taking molecular clock dates at face value. Usually molecular clock dates are too old, but in this case they may be too young.
Yeah, yeah. No, Andy, you're right. There aren't any fossils that we use to calibrate these trees, and our estimates have come from estimates of rate, and that has been done in a number of papers, starting with some of Andy Wolf's work, and then several of Dave's students have tackled this question too. And actually, I'm glad you brought this up. I do think that our original, or the estimate that I said in the beginning, the 4 to 6 is a bit younger than the one that Malia estimates as part of her work with this 353. I don't think it goes much later though. I think she might have pushed it back maybe a million years or so, but it's still a relatively, each of these separate studies have all definitely placed it within the last 7 million years, if not more recent. To your point, yes, we should be careful with estimates like that. The other thing too is that I do think a lot of these hybridization events have really been happening a lot in the last million years, like in even more recently, thinking about like the Pleistocene and like the glacial like turnover that's been happening in that time frame. And so yeah.
So Tom Chester had a comment. He says, it sure would be nice if we had some idea of where these species lived during the wet phase of the ice ages, but I suspect only the molecular something could give us clues to that. If it is possible to tease that out of the gene flow. I'll look forward to your paper discussing this.
Yeah. It is really cool thinking about how these sort of compartments have been moving around, and I often think about them in geographic space too, like where are they going when they come back together, and then when they diverge again. And this makes me think, Ian, back to your question about Exerta, and it is still very confounding thinking about these wide-ranging, these species that cross compartments, and yet and therefore are engaging with different gene pools, and yet still maintain a morphological consistency. It's sort of mind-boggling, and I really do look forward to having some of these data sets that can actually help us begin to test, where in the genome are these morphological traits that we sort of rely on to distinguish taxa, and what is, where are these morphologies coming from? Yeah.
Okay, Sarah, I think we should wrap it up. I'm not sure we're going to have a July seminar. Check the website to see, or you'll get an announcement. If you're a member, you'll get an email.